Hi everyone, I'm Ameera Nasheera. Today, we are going to talk about the metabolomics role in negative energy balance and the apoptosis of dairy cows. My team members are Siti, Khairunnisa, Sharifah, Haziqah, and Lim Xiang Chuin. Metabolomics is the study of metabolic mechanisms that are done by measuring metabolic products in biological specimens as a whole and by elucidating the underlying chemical process. Negative energy balance occurs when the energy used by the cow is higher than the energy intake from the dry matter. It usually occurs during the lactation week of cows to produce a large amount of milk. If this stage prolongs, drastic decrease in cow body mass will occur and cause disease to the cows. Energy balance can be achieved by modifying the food intake and physical activities for the cows.
Metabolomic changes in milk sample in relation to energy balance can be studied using liquid chromatography, mass spectrometer, and proton nuclear magnetic resonance (NMR). This metabolomic technique helps to detect and quantify the metabolite present in milk produced which are responsible for the negative energy balance in cow. The technique started with the milk sampling and analysis using spectroscopy method. Firstly, the milk sample of 87 dairy cows were collected during the lactation week 2. Then, the milk sample turned to room temperature and the fat layer was removed by centrifugations. Milk serum is mixed with the phosphate buffer and these samples are filtered to remove the protein by centrifugations. The sample was run by NMR spectroscopy, which is the spectra is obtained. Next, the same sample were used for the analysis with the triple quadruple mass spectrometer using the PFPP method.
LC-MS milk spectra, 97 metabolites were targeted and only 27 milk metabolites were detected. However, in NMR spectra, the lactose has dominated the region from 3.52 to 3.95 ppm, and these mask the signals for other metabolites in this region. And finally, 35 milk metabolites were detected in NMR. The integration between LC-MS and NMR gives the 55 milk metabolites were detected in the milk sample of the 87 dairy cows in the lactation week 2. The cows had also large difference in energy from negative 500 to 500 kJ per kg because of the large difference in the metabolites present in the milk sample. So from the research, we can conclude that 15 metabolites were positively correlated, showing low amount of metabolites, and the 26 metabolites were negatively correlated, showing higher amount of milk metabolites. And this imbalance in the correlation gives the effect in the negative energy balance of the cows.
One of the effects of severe negative energy balance is apoptosis and cellular proliferation. Apoptosis is a form of programmed cell death or cellular suicide. Apoptosis and cellular proliferation occurs in cows with negative energy balance with increases in the synthesis of nucleic acid, cell membrane phospholipids, protein glycosylation, one-carbon metabolism, and lipid metabolism. When there is insufficient energy, the metabolic system will use up fats, proteins, as well as cellular components to generate energy. This causes cell death in cow and releases the cellular content into the milk. For example, lipids are the main source of energy. During the energy deficiency, the fat below the tissue will be metabolized to generate energy itself. This in turn leads to the loss of body weight. When it continues for a longer time, it will cause diseases. Rapid cell renewal also occurs, increasing the numbers of memory epithelial cells critical for milk production. Therefore, there is a relationship between the apoptosis and cell proliferation and negative energy balance of the cows.